Very good afternoon, students. So before we start the session, okay, I would like to launch some polls for technical check for voice clarity, as well as if you can see the board clearly. Okay, please. Okay, hold on a second. So we don't have polls. So unfortunately, we don't have the polls today. So can you just put in your chat box if my voice is clear as well as if the screen is, visible? Can you see the screen that I've shared? Okay, so we had some technical issues. So now is it visible? Is the screen visible right now? Just let me know in the chat box if my voice is clear or the screen is visible so that we can start our session as soon as possible. Okay. Okay, students. So today we are going to start a new topic, that is biomolecules, right? Which is the term to first chapter. So today we are going to have an interesting session because in most of the biology that we have studied, so we talk about plant biology, we talk about animal biology, but today we have a very unique topic to talk about, that is biomolecules, where we deal with chemistry aspects of biology, and we understand how significant they are in our living system. So please, you can let me know if you have sessions right now. Okay, so initially, like when we talk about biomolecules, so all the living organisms are made up of basic inorganic elements. For example, hydrogen, carbon, sulfur, phosphor, potassium, okay, sodium, calcium, and magnesium, and all of these things. So when we look at a living organism as a whole, so we would not recognize it. So we will think that we are all made up of tissues, we are made up of cells. Okay, so as we go deeper and deeper into the structure of cell, okay, we talk about cell membrane, we talk about cell wall, and we talk about other cell organelles and proteins that are present within the cell. So when we study the chemical aspects of these cells or all the living organisms, so all of us are made up of very, very basic inorganic compounds. Okay, so that is what the study of these basic inorganic compounds and understanding, okay, the whole of it, like how it is contributing. For example, if I say carbon and its compounds, like how it benefits in constructing certain elements or compounds that make up certain parts of your living system. Okay, so like if you have to study a living system, okay, or a living tissue, and if we have to understand the biomolecular concept or biochemical concept of these living tissues, so we have to have a certain methodology for it. So we have two ways in which we can study, okay, a living system, a living tissue, okay, to understand what it is made up of. Okay, so initially what we can do is we can take any of the living tissues from, for example, a chicken liver you can take, okay, or you can take some plant tissues, okay, soft plant tissues, okay, or some vegetables you can take, and then you can try to grind it very well using this, okay, trichloroacetic acid. Okay, when we use trichloroacetic acid, now what happens is usually the cells and the tissues are quite attached to each other, right? They're very highly integrated with each other, and there is a proper gluing between the cells or gumming between the cells. So for the tissues to just disintegrate from each other, or unstitch from each other, so we use trichloroacetic acid. So once we use trichloroacetic acid, now the tissues will become loose, and so as the cells. So once the cells become loose, we use this mortar and pestle. Okay, so we use mortar and pestle and start grinding these liver tissues, or you can talk about vegetables, or some leaves or something like that, any living tissues. So once we grind it, it's gonna become a very, very, okay, thick paste. So once we have this very fine paste, okay, now what we have to do is we have to pass this fine paste, okay, through a, for example, cotton cloth, or you can call it as cheesecloth. Okay, so cotton cloth or cheesecloth, what it will do, it will separate all these, okay, solid portions, and it will separate the liquid portions, right? So just like how you filter the tea, okay, so all the tea leaves will be in the filter, so and the rest of the tea will fall in the tumbler, right? So similarly, when we filter this, okay, the ground tissue, now what we get is we get a complete filtrate. So once we analyze this filtrate, we get two portions of the filtrate. Okay, for example, if I take, okay, let me say, if I take, hold on, let me change some ink so that we don't have a problem. Okay, so let us take that. I take a test tube. Okay, I take a test tube and I use this filtrate. If I use this filtrate, I will get, okay, two different layers. Okay, I will get two different layers. So one layer of this particular filtrate will be acid soluble pool. Okay, that means it can dissolve in acid, and other part of your filtrate will not be soluble in acid, though that we call it as acid insoluble fraction or acid insoluble pool. Now the composition of this acid soluble and acid insoluble pool will give us a very brief idea about what these molecules might be. So when we talk about acid soluble pool, so this particular filtrate will contain, okay, all the bio-micromolecules. Okay, the name itself suggests bio is living, micro is which is present in, okay, I mean, like the size of these particles is very, very small. Okay, so bio-micromolecular portion. So these micromolecules, their weight is usually less than 1,000 Dalton. So usually we take a measurement up to 800 Daltons. Okay, so if it is below 800 Daltons, so then we call it as bio-micromolecules. Whereas the acid insoluble fraction, what we talk about macromolecules. Okay, so don't worry about what is micromolecules and macromolecules, because in this entire chapter we are going to discuss in detail about this, okay, particular micromolecules or macromolecules. So when it comes to acid insoluble fraction, okay, it contains majority of these macromolecules, and these macromolecules are heavyweight. Okay, they are quite larger in their density, so they are usually more than 1,000 Daltons. So what did we do? We wanted to analyze what these living tissues are made up of. So we extracted the living tissue and we started treating them with trichloroacetic acid, and then we got a filtrate, and through the filtrate we analyzed that there are two portions to the filtrate. One portion of the filtrate we have acid soluble pool, and another portion of the filtrate we have acid insoluble pool. So we understood that acid soluble has micromolecules, and acid insoluble have macromolecules. Okay, so when we look at our surroundings, right? So we just conducted an experiment and we understood that all living tissues are made up of these organic or inorganic compounds. One fascinating thing that we can learn from our living environment is our organisms, all the organisms, including us, we have this tremendous capacity. Okay, we have evolved so well, okay, to construct and destruct, certain things. For example, when we are born as a single cell, we keep on constructing, right? Our body, adding more and more cells. So when we look at our cells are made up of certain basic biomolecules such as lipids, such as proteins, nucleic acids, okay, so etc. So when we look at these biomolecules which make up your cell, okay, all of them are made up of very basic elements like your hydrogen, carbon, water, like oxygen, nitrogen, sulfur, potassium, okay, calcium, magnesium, iron. So for example, take your blood. Your blood contains, the heme protein contains what? It contains iron, isn't it? Okay, we have iron in our blood. So this is also one of the elements that are present in our nature, isn't it? So for example, your bones, it contains calcium. Your cells contain sodium as well as potassium for signaling, right? So most of the cells contain oxygen because it is very much essential for respiration, right? So your lipids contains carbon and hydrogen, right? And including oxygen. So your proteins contain sulfur, okay? Proteins contain sulfur. So all our biomolecules are made up of these basic elements which are present in the nature. So through the process of metabolism, right? So through the process of, sorry, I'll just raise this. Hold on. Excuse me. Okay, through the process of metabolism, okay. So the process of metabolism, what do? We construct or destruct, okay, for our benefits. So we construct proteins, we construct nucleic acids, okay, we construct phospholipids, or we construct, okay, so a certain cell organelles, certain tissues. So all this is coming from very basic elements that are present in our environment. So the food we eat, okay, are broken down into simpler elements, and the simpler elements or compounds are again used for the reconstruction of specific things that are required for an individual organism. So if we just understand the story of construction and destruction mechanism of a living system, then we can just simply understand that all living organisms are made up of non-living elements. Okay, the non-living elements make up a living system, right? So it is such a fascinating thing to even accept this fact. Okay, we just think that we are living, but when you look at the deeper parts of our living system, it is all like what is present in your inanimate or non-living earth, like your soil or the seawater, okay, or the mountains. It's just like that. So if I just analyze what's the difference between the earth and the human body, the earth and the human body share so many different elements which are very, very similar in both the cases, or same. For example, take for example hydrogen. Okay, wait, give me a second. Okay, let me change again. Okay. Yeah, if I take an example of hydrogen, right? So hydrogen in the Earth's crust contains 0.14%, whereas in human body it is composed of up to 0.5% of the total weight of the human body is made up of hydrogen. So take for example carbon. So most of our sugar like glucose, right? And when we talk about the lipids, when we talk about sterols, so many so many of our biomolecules contain these carbon compounds. Okay, so if you look at the Earth's crust, even though the carbon content is so less in the Earth's crust, that is just 0.03% of the Earth's crust, but when we compare it to the human body shows fascinating amounts of carbon content, that is up to 18.5% of the human body is made up of carbon. So take an example of oxygen. So oxygen is one of the highest elements that are present in the human body. So up to 65% of the human body, okay, among the total weight, 65% of the elements that are present is made up of oxygen. So we have nitrogen, sulfur, sodium, calcium, magnesium, and one more element is missing. I will add it here. So that is potassium, right? So potassium is also one of the major elements that are present in the human body. So all these elements that are similar to the Earth's crust, okay, they are used up in different forms for the construction of, okay, micromolecules or macromolecules, so that they become one functional molecules for the construction of a living cell. Okay, so are you following me? So if you have understood, please let me know, or if you have doubts, you can just ask me. Okay, so it is very essential for us to understand what is the total weight and how much is the composition of certain elements. So if I give you a question, okay, between oxygen, carbon, okay, then I take nitrogen, then I take potassium, then I take calcium, and then sodium, and then ask you to just calculate, okay, and tell me which is the highest elements, for example, in increasing order or in decreasing order. If I ask a question, so you should be able to memorize this, okay, just like this. Okay, you can tell, if I ask you which is the highest element that is present in the human body, so you should be immediately, you should get the answer like it is oxygen, which is made up of 65%. Okay, next the least is potassium, and then comes sodium, okay, and then comes calcium. So similarly, as we keep on studying these elements, we also come across certain elements that are not present in the human body. For example, if I take copper, right, if I take copper is not present in human body, right? So iron is present in human body because iron is used for the, right, for the construction of red blood cells or heme protein, whereas copper is not much used in our in our living system. So copper is absent. Did you understand that all living organism is made up of non-living elements? And this is the comparative analysis of your non-living and living system. So let us start our topic by understanding what is bio-micromolecule. Okay, we'll start studying what are the different bio-micromolecules and what are the properties of these bio-micromolecules. Okay, so if we have to consider about bio-micromolecules, as I said, any molecule which is, okay, between 18 to 800 Daltons or within 800 Daltons, so we call this as biomicromolecules. And mostly biomicromolecules, as I said, they are found in what pool? Acid soluble pool, right? So usually when we talk about acid soluble pool, so all these ingredients that are found in the acid soluble pool, when you look at in the living system, so if I have to just understand, so if I just say that acid soluble pool is present, and then you have a question, where is this acid soluble pool or the micro molecules are present in a living cell? So in a living cell, basically all these micromolecules are present in the cytoplasmic constituents. Okay, so they basically occupy, okay, or they basically are present in the cytoplasm of the cell. So coming to the different types of micromolecules, okay, so we have different types. That is, first one is amino acids, okay, second type is lipids, then we have sugars, and then we have nitrogenous bases. So these are 4 different types of micromolecules. So we will study individually about each micromolecules and we will start understanding their properties. Okay, so coming to the first micromolecule is amino acid. So if I ask you what are amino acids, okay, you might have different opinions about it. Why? Because some of you know the name amino acid or the word amino acid. Some of you really know that amino acids are the basic, okay, units of protein construction. So students, like if you just look at the function, for example, how the living system functions. So yesterday also, while we are studying cell biology, I told that all the information of the cell is present within the nucleus in the form of DNA, right? All the information is present in the form of DNA. And then the information is passed to the cell for the, to just do certain job. Okay, it has to tell the cell, you have to construct this, or you have to destroy this, or you have to create this, okay, or you have to transport this. So all these messages that the cells perform, all the functions that the cell perform, all the messages for these functions is present in mRNA or just RNA. Okay, so the messages from the mRNA are further used for the construction of what? Proteins, right? So when the protein is constructed, so all, for example, if I take 98% of your all biological functions, okay, all the functions, all the biological functions that you perform, okay, like your movement, your metabolism, okay, or your transportation, or your stimulus, your hormones, okay, or your enzymes, all of these activities or all the functions are essentially carried out by proteins. That is, their function is up to 98% of all biological functions are made by proteins. So if I look at proteins and then ask a question, what are proteins made up of? So the proteins are, polypeptide chains. Okay, so we have proteins. Okay, I'll just write it here. Protein. If I just open the protein, so proteins are made up of different chains and we call it as poly. Poly means many, and then it is peptide. Okay, polypeptide chains, right? They're made up of polypeptide chains. So if I take one chain and then look at the chain, what do I get? I will get that the protein is an assembly of, okay, it will be like a chain. Okay, it's a chain molecule or a linear chain molecule, we can call it as. Okay, it is an assembly of different amino acids. So several amino acids. So for example, there are around 20 different types of amino acids, okay, in our living system. I mean, like according to the total number of amino acids present is around 20 different types of amino acids that living organisms use. Okay, so among these 20 different types of amino acids that are present, okay, so we use these amino acids in different ways, in different combinations to construct a specific protein. Okay, so once we assemble this, for example, if I take one amino acid and then join with another amino acid and go on with another and go on and go on. So now this series of amino acids that are, just connected with each other, or a chain of amino acids, will give us what? Polypeptide chain. And one or two or three or four or more than four chains, when they combine, they form this structurally, okay, and functionally, a structural and functional biomolecule that we call it as proteins. Okay, so we understood that amino acids are the basic unit of polypeptide chains and proteins. So let us see the chemical structure of an amino acid. So if I have to understand the chemical structure of an amino acid, so it's very easy for you to, just understand this. How do we do it? For example, if I ask you to write a structure for methane, okay, CH4, let us say, what would you do? You'll write one carbon atom in the center, and then you add 4 hydrogen atoms in 4 different directions, or you just do it like this, right? Sorry. Okay, hold on. So for example, you will write like this. So this is the structure of CH4 you will do. So you just remember this CH4 structure, and then what we can do is, let us leave this hydrogen here and let us go for the rest 3 of the hydrogens. Okay, so what I will do, I will remove this hydrogen. Okay, I will remove this hydrogen, and then I will add a carboxylic group. Okay, COOH group I will add by taking out one hydrogen atom. Then again, what I will do, I will remove one more hydrogen atom, and then I will add an amine group, okay, or amino group. Okay, so next, what will I do? I will remove one more hydrogen atom. I will remove one more hydrogen atom, and then I will add a functional group, that is R group. Okay, so if I construct this, okay, what did I do? I took a CH4 to understand the structure of amino acid. So I wrote one carbon in the center, and there were like 4 hydrogen atom. I spared one hydrogen atom, and then the rest of the 3 hydrogen atom were substituted by one carboxylic group. Okay, and then we have an amino group, and then we have a functional group, that is R group. So in total, this will be the general structure of amino acid. Okay, are you able to understand, students? Okay, you can just put it in the chat box if you have any doubts, so we can sort the doubts. Okay, fine. So the general structure of amino acid is very, very important for us to understand. So why we have to study this general structure? So I told you amino acids are, even though some of the amino acid can, function independently or individually, yet almost all the amino acids are used up for the construction of complex proteins. So if I have to construct complex protein, first I have to understand the structure. How do I add two different amino acids together to form one protein? Okay. So what will happen if I see an amino acid? I told you that at one part of your amino acid is your amino group, right? And then another part of your amino acid is your carboxylic group, right? So since we have an amino group, that is why we have amino, amino, okay? Then we have carboxylic acid. That's why we have taken acid from this carboxylic group and then we have added here. So that is why amino acid, okay? Need to get it? So if I see a amino acid, what I can find is, so in a general structure of amino acid, so I have an amino group, okay, which has a nitrogen here. So I will call it as N-terminal, okay, N-terminal. And then on the other side of the amino acid, I have a carbon atom or carboxylic group where I have C-terminal, okay. So I have two N's, one is N-terminal and then I have C-terminal. So now what happens is, so if I have an amino acid 1 and then I have an amino acid 2, the N-terminus of the first amino acid will combine with the C-terminus of the second amino acid through a linkage, and this linkage we call it as peptide linkage, okay. So through peptide linkage, now what happens? A polypeptide chain starts forming. So this is about the general structure of amino acid. So did you understand, people? Okay. If you have any doubts with the structure of amino acid, you can let me know. We can discuss about this. Okay, fine. So once we understand the general structure of amino acid, and I told you how this structure helps in construction of polypeptide chain, another important factor about amino acid is its chemical nature. Okay, so every chemical, okay, every molecule that is present in our universe, they exhibit certain qualities. We call them physical properties and chemical properties, right? So when we talk about chemical properties, we talk about acidic, we talk about neutral, we talk about basic, we talk about polar, we talk about non-polar, etc., etc., isn't it? So we talk about different types of chemical characteristics. Okay, so similarly, amino acids also exhibit, okay, a unique type of chemical characteristics according to the presence of their functional group. Okay, so I will give you more examples. Let us see. Okay, so if I see, hold on. So if I see an amino acid based on the presence of their functional group, right? Based on the presence of their R groups. For example, in glycine is a type of amino acid and the functional group is H, right? Similarly, you have alanine is a type of amino acid and their functional group is CH3. Okay, and then we have serine is another type of amino acid and then their functional group is CH2. Now, if you look at the structure of these different amino acids, some of the things are very common. So what do we have? We have a C, we have H, we have N-terminal, we have C-terminal, and look at this functional group, it is H. And look at the functional group here. So the functional group is here, correct? And look at the functional group here. The functional group is, NH2 and then OH. Yeah, we have OH as the functional group. You can see here, right? So we can clearly understand that based on the presence of the functional group, now the structure of this amino acid has also changed. Now with the change in the structure and the presence of this R group, they also show a different types of what? Chemical properties. Now these chemical properties are highly essential, okay, in determining what type of protein and what is the nature of the protein, what is the structure of the protein that is going to be created. Okay. So before we go in detail about the different types of amino acids, let us talk about what are amino acids and what are the different types we have. So here we have, according to the different functional groups, we can categorize amino acids as 20 different types of amino acids are used, okay, for protein synthesis. Okay. So all these amino acids that are used, okay, for example, if I take, okay, an example of here, so here you can see here we have glycine. Okay, you have your entire list of amino acid here. We have glycine, we have alanine, valine, leucine, methionine, isoleucine, proline. And look at the characteristics of this amino acid. So the characteristic of this amino acid is, okay, non-polar aliphatic R groups. Okay, they are non-polar aliphatic R groups. That means, what is non-polar, right? So similarly, we have polar R groups, and then we have positively charged R group, and then we have negatively charged R group, and then we have aromatic R groups. Okay. So among this list of amino acids that we talk about, polar, non-polar, positive, negative, and neutral, one of the important groups that we have to remember until your NEET examination is your positively charged R groups. So why positively charged R groups that are essential for us to be memorized? There is a specific reason for it. Okay, I will tell you why. Because when you talk about DNA and all that, so you will really study about this positively charged amino acid, and many times the structures or the questions have been asked. So when we take an example of this positively charged R groups, so we have three different types of amino acid. That is, one is lysine, and the second one is arginine. Hold on, let me change the ink. Okay. So we have lysine, arginine, and then we have histidine. So these are the amino acid that show, okay, positive charge. And this positive charge is very important for the packing of DNA. Okay. So we'll talk about this later when we talk about nucleic acids and all. So this is about amino acids and their chemical characteristics. Okay. So let us go back and study in detail. So as I said, there are 20 different types of amino acid based on their R groups. They have their particular chemical characteristics, right? So when you see amino acids are very, very important for us on a daily basis. So usually we get amino acid from food, or we can synthesize the amino acid by ourselves, like our body can make our own amino acids. So based on this, we have two important classes of amino acid based on our requirements. So first we have is essential amino acid, and then second one we have is non-essential amino acid. Now, what is essential amino acid? Essential amino acid is the group of amino acids where your body cannot synthesize it, right? I cannot produce this amino acid. So the only source for this amino acid for me should come from the food you eat. Okay. So the food you take will have the certain amino acids which your body cannot produce. So the amino acids which your body cannot produce, and you get those amino acids through the food you eat, those amino acids are called as essential amino acids. Okay. And the example for essential amino acids are lysine, leucine, isoleucine, and tryptophan. All right. So whereas on the contrary, so we have non-essential amino acids. In non-essential amino acids, what do we have? We have glycine, alanine, serine, and arginine. So what are non-essential amino acids? Our body can synthesize on its own. Okay. So there is no dependency for these amino acids. Okay. So this is about amino acids based on based on the requirements of a body, right? So our body requires this, or it can produce this. Based on the requirement, we can categorize them into two different classes. Okay. So now the question for you is, list down what is the total number of, okay, I'll just write it here. Okay. What is the total number of essential amino acids, number of total amino acids in humans. Okay, let's talk about humans only. because it depends from one organism to another organisms, like what is the type of amino acids and what is the essentiality of this amino acid. So if we talk humans as an example, so you can understand how many different types are there. All right. So when you look at the structure of amino acid, there is one specific or peculiar character about this amino acid. So what did I tell you? When you see the general structure of amino acid, I told that they have two terminal. One is an N terminal, other one it is a C terminal. And N terminal is a portion of your amino acid where you have an amino group, right? So here, so hold on. So when we talk about this amino group, so what charge does it present? It shows the charge that is positive charge. The N terminal of your amino acid, where the amino group is present, there you have a positive charge. In the same, in the same structure, if you see the other portion of it, you have another terminal, which we call it as a C terminal or the carboxylic acid terminal, where there is an essential charge of negative charges present. Okay. So usually some of the amino acids, what they show is if they are present, okay, if they are present as, for example, NH3. Okay, here is the structure. We'll use this structure to study. For example, we have NH3 here. So there is one positively dominant charge is there in a particular amino acid. So this we call it as cationic amino acids. All right. So similarly, we have anionic amino acids where you have a high negative charge at the carboxylic end. In most cases, what happens is amino acids show a characteristic where they have a balanced, okay, positive charge at one terminal, and then you have a negative charge at the other terminal. So amino acids essentially are what type of ions? They are Zwitter ions. What are Zwitter ions? Zwitter ions are compounds where in the same chemical structure, you have both positive as well as negative charge. So your amino acid is a Zwitter ion. All right. So did you understand about this charge of amino acids? Can we move further? Okay, students, like you can just put it in the chat box if you have any doubts, okay, or if you have any problems. Okay, it will be very helpful and we can make the class interesting. So remember this, table and try to summarize, memorize these structures, okay, as much as possible. So when you solve these questions, you can see some of the questions are coming from glycine and alanine and valine, importantly, okay. And then you have the questions which are asked from serine and lysine, arginine, histidine. Okay, these amino acids are specifically targeted in the examination. And the reason is very simple, because, for example, if I take valine, we study valine in second year in some of the genetic disorders. Okay, if we talk about lysine, arginine, and histidine, we relate them to the packing of DNA or genetic material. So whatever the examples, okay, of proteins and a specific quality of a protein is talked about, so we really have to go back to the amino acids that they have mentioned. Okay, so these are certain amino acids that are usually mentioned in your textbook, and please concentrate on their structures. Okay, so fine. And our next micromolecule that we have to talk about is lipids. So what are lipids? Lipids are basically water insoluble compounds. So essentially they are made up of three basic elements. One is carbon, other one is hydrogen, and then you have oxygen. Okay, so you have hydrogen, carbon, and oxygen containing compounds and which are water insoluble, and these we call it as lipids. So usually when we talk about lipids, so why do we require lipids? So what I told you, why we require protein for 98% of the biological functions that a living organism performs. Whereas what about lipids? What is the use of lipids that we have? Okay, so when we talk about lipids, usually we come across certain terms called as fat, right? We come across cholesterol. Okay, sorry. Okay, I'll just write as sterol. Okay, we come across sterol, and then we come across oils. Okay, we come across waxes, right? So there are so many different derivatives of these lipids. But when you see the function or the importance of lipids in a living organism, for example, you can take bear or polar bear. Polar bear lives in Antarctica where it is very, very icy, isn't it? So when you see the capacity or the ability of these whales or seals or polar bears or penguins to survive in these extreme cold conditions, so for them, the blanket from this extreme cold is coming because of the presence of high lipids in their body. Even for you, if you do not have sufficient lipid volume in your body, so then you will have problems where you cannot control, cold. Immediately you feel like very cold with the small changes in the environment. Okay, so lipids provide us what? It will provide insulation. So it will protect us from heat, cold. Okay, it will protect us from cold, insulation from cold. Next one, they are very much useful for the construction of plasma membrane. So your plasma membrane is what? It is a lipid bilayer, isn't it? So it is made up of phospholipids. Okay, take for example testosterone or estrogen. Okay, so we are talking about certain hormones which are steroidal based hormones. Okay, all these sex hormones are steroid based hormones which are again lipids. Okay, so lipids have a very vast application with respect to living system, including birds. For example, the birds produces this waxes. Okay, they produce these oils to protect from extreme cold or something like that. All right, so all these things will help the living organism, okay, to provide certain things. Okay, so let us see what are the lipids. So lipids are basically categorized into three different types. One is simple lipids, and then you have compound lipids, and then you have derived lipids. So we will study independently about different types of lipids, and we will try to learn about their characteristics. Okay, so when it comes to simple lipids, right? So usually the simple lipid, we will talk about fats and oils. Okay, so I will just write it as fats and oils. So is one of the simple lipids. Okay, so when we talk about fats and oils, usually fats, okay, if I just tell as fat, so what are these fats? So the fats are usually made up of two important components. So one is, the fat is composed of one component called as fatty acid component, and the second component is called as glycerol component. Okay, so based on their combination, so we have different types of simple lipids. For example, I will give you an example of how this structure is working. Look at here. I told you the lipid has two important components. Okay, especially when it comes to fats. Okay, especially when it comes to fats, the fats contain two ingredients, right? So the first ingredient we will talk about is glycerol. So the glycerol is an alcohol derivative where you have three carbon atoms: first carbon atom, second carbon atom, and third carbon atom. So we have three carbon atoms, and then overall we have 5 hydrogen atoms, and then we have 3 OH. Okay, we have 3 OH groups. Okay, 1, 2, and 3. So this structure is called as a glycerol. So for the glycerol, okay, a long chain of hydrocarbons are attached. So let us see what happens here. So you can see here in this slide, okay, in this slide very easily. So you can see we have glycerol here, right? So we have glycerol here. So to this glycerol, now what is happening? Another additional group is attached. What is this group called as? So the first element we talked about is glycerol, and the second element of this fat is called as fatty acid. Okay, it is called as fatty acid. Now what happens will, for this fatty acid, what are fatty acids? These are long chain. We have carbon and we have hydrogen, isn't it? So they are what? They are long chain long chain hydrocarbons, right? So they are long chain hydrocarbons. So when we talk about long chain hydrocarbons, Usually the number of carbon atom can go from, okay, 8 to 38. So usually in our living system, we have around 8 to 18 or 19, okay. 8 to 18 or 19. What are we talking about? The number of carbon atoms. You can see here 1, 2, 3, 4, 5, 6, 7, 8, and so on. So is a number of carbon atoms, okay, that represents in a fatty acid chain. Now, why do we call it as fatty acid? We call it as fatty acid because at one end of this hydrocarbon chain, we have what group here? Can you see here? We have COOH group. So that is what carboxylic group is present, which gives the acidic nature, right? So excuse me. So time, seriously, okay, students. Thank you for letting me know. I thought it is still 5 o'clock. Okay. Usually it is from 3:50 to 4:50, isn't it? It's not like that. Anyways, before we end the session, did you understand today's class? And we can continue tomorrow also. So just give me a quick response so that we can end the session right away. Students?